Okay. The cell cycle refers to the cell's life. You may remember learning things like the life cycle of an insect, where you've got egg, larva, pupa, adult, and then the adults lay new eggs and it starts over again. For a cell, the process is a little simpler. You have a cell, and it divides to make two new cells. Then each of those can come back around and divide again. We call the starting cell a parent cell, and the two new ones daughter cells. This cell's life starts once it's completed splitting off from its parent cell, and it ends once it finishes splitting into two daughter cells. There's a little more detail within each cell's life cycle. It spends most of it in what's called interphase. They called it that meaning between divisions. That's the three large pie sections here. Interphase is divided into G1, S, and G2. S stands for synthesis, and that's when the DNA is replicated or copied so that each daughter cell can have a full set. In the G phases for growth, the cell is growing, making proteins, making more organelles if it needs them, and so on. And then the green section here is what's called mitosis. That's where the cell divides up that copied DNA into two separate nuclei. And at the same time, cytokinesis is dividing the cell itself into two separate cells. If we were to compare this to a human life cycle, cells start in G1 and they're already adults. They don't have to grow up. Once they're born, they're ready to do their job. Some cells are going to stay in G1 forever and never divide. Okay, so they're the ones who decide not to have kids and just focus on their career. Or they just don't want kids. Okay. So G1, the cell's doing whatever it's going to do. If it's going to divide, it'll pause and go into the S phase. And that's like ovulation and conception. You take a moment, you start on that path towards dividing. G2 would be, you're building the baby for the cell. It's building proteins that it needs in order to divide. Then mitosis is when you go on maternity leave and the only thing you're doing is having that baby. All the other jobs just sit by the wayside. Now, the big difference is that when the cell divides, it splits into two equal portions instead of budding off another small one and keeping its original identity. So instead of, let's say, Lacey decides to have a baby, and then we have Lacey and Lisa. If Lacey were a cell, she would instead split in half. There's no Lacey anymore, there's Lisa and Lucy. Now, we don't know if that's really what it feels like for the cells, or if they have any sense of identity, but that's how we described it, as these are brand new cells, and that one's gone. Mitosis, dividing the nucleus, has 4 stages to it: prophase, metaphase, anaphase, telophase. You add interphase to this for the whole cell cycle, you have a pronounceable acronym, IPMAT. here's the overview of the stages. And now I got to figure out how I can draw them over there and still record the slides over here. I think what I'll do is I'll go through the slides and then I'll move the camera and go through the phases again, drawing them. A couple of definitions related to the DNA. Okay. We mentioned the chromatin, the mass of DNA, and some organizing proteins in the nucleus. Each piece of DNA is a chromosome. Humans have 46 of them. So there are 46 separate pieces of DNA in each of your nuclei. A chromatid is each copy of a chromosome. So in the S phase of interphase, all those chromosomes are copied. The two copies are kept together. So each chromosome has two chromatids. Prophase, think prepare. Now, mitosis is kind of like splitting up some copies. In interphase S, it's like you send your 46-page document to the copy center. And instead of sending you 2 copies of pages 1 through 46, they send you 2 copies of page 1 stapled together, and 2 copies of page 2 stapled together, and so on. And so the cell has to go through and organize, collate everything. Now, where your document would be copied on nice, neat sheets of paper, the chromosomes are stretched out like pieces of string. So first they have to get crunched up into neat pieces so that they can be moved around without getting tangled. We call that condensing. And that's when the chromosomes become visible as separate pieces. We draw them as X's because they each have the 2 chromatids held together. When you look in the microscope, you won't always be able to see the X's, but you'll see distinct pieces instead of just a dark nucleus. Second, we got those copies from the copy shop in an envelope, and we can't deal with them inside the envelope, so we need to take them out. The envelope for the cell is the nuclear envelope. So the nuclear envelope comes apart in prophase. And then the last thing, now one place in the textbook describes this part as being metaphase, but almost everywhere else describes this as prophase, so this is where I'm putting it. The centrosomes, that little pair of cylinders, you make a second pair in G2. They are going to move to opposite ends of the cell. We call those the poles. And they're going to extend spindle fibers, which are microtubules, and attach to each chromosome. So this is like, the centrosomes are 2 people who are going to help organize those documents, and they reach out a hand and grab each page. They have to have 46 hands, and that's your spindle fibers. So that's shown here with these little strands connecting to the chromosomes. Questions on prophase? Metaphase, think M for middle. The chromosomes line up right along the middle of the cell. Because we call the ends the poles, we call this the equator. You'll also see it called the metaphase plate. So this is kind of like the centrosomes go, okay, let's line this up and make sure that we both have one of each copy. The metaphase is pretty short. It's just a nice, neat lineup, and now we're ready to separate the chromatids. That is anaphase. This is the same ana as in anatomy, cut up or cut apart. Now it's the phase where they go apart, and that's the chromatids. So we pull out the staple, we move 1 copy of each toward the poles. Here on the diagram, or on the photo, you can see two separate clumps of chromosomes. And then telophase, telo means end, think T like a terminal. Basically, telophase reverses prophase. New nuclear envelopes form around each set of chromosomes. The chromosomes are going to decondense, so they unravel out into strands of chromatin again. Now, at the same time, cytokinesis is happening, dividing the cell. They didn't show it in the drawing, but you can see it happening here in the cell, where it's pinching in from the sides. Cytokinesis, we divide the whole cell. This is a job for microfilaments, with microtubules moving the chromosomes. The microfilaments are going to form a ring at the equator of the cell, and kind of like a boa constrictor, they pinch in, pull the membrane closer and closer together until it touches. And then there's just this little bit holding it together, and it can split apart. Now, sometimes you can see cytokinesis beginning in anaphase. It's usually clearest in telophase, and when cytokinesis finishes, that's when we consider it the end of mitosis, and now we have two new daughter cells in interphase. There are a lot of mitosis animations on YouTube. If you like learning from videos, you might find it useful to go through and do a search. What we'll do is we'll pause the video, we'll start a new video for me to draw our way through the phases.